Thanks everyone for the invitation and a very exciting day that I could attend with many different faces from your group, Christian, and from Robert's team. And I learned a lot. It was an exciting journey with your different topics in population genomics. And yeah, as you rightly introduced, I come from population genomics, experimental population genomics in Markus Penninger's group. But today I would like to talk about the science that I have been developing in the past 4 years here at University of Cologne. And of course, this should focus on defining my own research field and developing my own research topics, which circulate in this title, Understanding Ecological Impact on Genome Evolution. And I promised to talk a bit about rivers and about deserts. And the reason for that is that I would like to present two projects that are ongoing in my group here in Cologne. So first of all, how can we actually understand ecological impact on genome evolution? We have to figure the fact that there's some interaction in ecology and evolution. Obviously, we know that the evolutionary timescale dates back to the ancient past, covering a very long timescale. And on the other hand, we know that ecology only covers parts of this timescale. But nevertheless, in the past decades, we have been learning a lot about the concepts of rapid evolution, and obviously the timescales of ecology and evolution do overlap. And this is actually this overlapping window in which we do have to expect that there's a certain degree of interaction between ecological and evolutionary processes, feedback, or producing and feedback towards each other. Potentially, we can also learn a lot about projections to future scenarios from understanding ecology and evolution in this window of interaction. And since I come from population genomics, obviously my target level of interest is the genomics world, and this is where my science nowadays circulates in the field of so-called ecological genomics, because we can actually span this time window of eco-evolutionary interactions with genomic data. This is, at least is my hypothesis. So how can we then measure eco-evolutionary interaction? On the one hand, you are all pretty much aware of factors driving genome evolution, which is the evolutionary forces as such. And on the other hand, we have the world of ecology that from my perspective, coming from molecular ecology, would mostly define upon the demography of particular species and populations, the environment as such, and of course, the mobility of biota in these environments, so dispersal patterns. And you see here that these two worlds come a bit separate next to each other. Nevertheless, I tried to make clear that there is some feedback between these different worlds. And the question is, how can we measure it? And how can we do this with genome data? So let's put all the ingredients we have on this scale, and we find that there's actually a lot of complementary aspects in the world of ecology and in the world of evolutionary biology. So when we observe for our evolutionary studies, population genomic studies, we usually work on the level of populations. We do investigate genetic diversity patterns or measure gene flow, or are interested in mutation as process for genome evolution. On the other hand, we of course can define ecology upon the community level of different species. We have to define the heterogeneity of the environment. As I said, it's important to understand dispersal patterns. And finally, there is the origin of new species as result from speciation processes. Whereas on the other hand, we have the origin of new alleles as the result of the mutation process. So there is some complementary aspects in these two worlds. And interesting enough, many of these we can directly measure from genome data. So we can get an idea on the community composition, applying molecular methods. We can measure dispersal or at least migration from genome data. We can get an idea on speciation, reading our genome data. Of course, we always focus on the population level for our population genomic studies, measuring genetic diversity, gene flow, and mutation, of course. So you see that genomics actually is a powerful tool to measure ecoevolutionary interaction. Unfortunately, not many people do this so far. So I asked myself, what can we finally read from genomes and learn about the ecological impact? And for this, I have chosen two alternative systems to ask more or less similar questions. And on the one hand, I have chosen a system that is super stable in its ecological condition. And on the other hand, it's a system that is as dynamic as you can imagine. So actually complementary systems also on this level, deserts and rivers. So let's start on the deserts part. I have had the opportunity to implement my research in an ongoing collaborative research cluster that is coordinated here from the Department of Geosciences in Cologne. And this cluster, the CSC, is called Earth Evolution at the Dry Limit, and it focuses on the evolution of land, landscape, and life in the Atacama Desert in Chile, which is a place pretty unique on our planet because it is the largest non-polar desert on our planet. It is the place where the Earth's surface is in its actual condition highly stable since the Miocene, so millions of years. It has been more or less unchanged. So we call it the oldest Earth surface on the world. And it is, of course, an extreme environment. There's hardly any precipitation. And as I already indicated, the ecological conditions are pretty stable since millions of years. So whatever has been evolving in these conditions has had relatively large amount of time to adapt to these conditions. And so I would hope that the time period of ecology and evolution to interact has been relatively long for this system. And so this is not only me, but we are a team together with my colleague, Dr. Philipp Schiffer here from the Institute with his Eminöta group, and a colleague from Stockholm, the National History Museum, and our two PhD students. And we do focus on the evolution of soil nematodes in the desert. So from my previous research, I come from the world of insects, like many of you guys, but I have learned a lot about it is important to have many individuals per sample, and you have to pass hundreds of generations, and studying with insects turned out to be very lengthy procedures in experimentation. So I decided, well, how about choosing another model? And nematodes obviously fulfill all these criteria. So I switched the model system and also the continent for my research. So why do we focus on nematodes? In general, they are super abundant. You can go wherever you want on the world and you will find nematodes in soils. They are crazy in their life traits. They can do something that is called cryptobiosis, which means that they can enter a state that is similar what you would know as dormancy. So basically they shut down their metabolism to zero, but yeah, to actually zero, because it's non-measurable. And this metabolic state has been defined or discovered already long ago. So this is an exciting life trait that allows these organisms to survive under super extreme conditions. They have been established as indicators in the world of ecology. So ecologists know a lot about nematodes, and for my world, focusing on genomes, I can say that they are genomically super exciting because they are hyper diverse on the genomic level, even though all the worms look the same, more or less. And there's a super high species richness expected. So we have a high taxonomic diversity as well. So yeah, for us, it was a very exciting model system. When we want to understand the impact of ecology on genome evolution, first of all, what we have to do is understanding the ecology, obviously, and this is what, many aspects of this presentation will now cover. So before we can really dive into these exciting genomic studies, obviously we have to build all these data at first before we can understand the impact on genomes. So what we did is doing an excessive sampling and also exploring different ecological variables, measuring richness on the level of families and genera, investigating different reproduction modes, which are exciting in these animals, analyzing feeding types, scenarios, etc. And then we did some model prediction to get an idea on the diversity patterns in the desert. And what we can find is that these extreme conditions, mostly driven by the factors aridity and elevation, do actually explain very nicely the patterns of diversity that we find in the desert in terms of richness, as you see here in this upper part of the charts, and the reproduction mode, so asexuality in the nematodes, which we can mostly predict with more with higher elevation and more severe aridity scenarios. So we can say something like the more diverse, the more extreme the habitat conditions, the more likely is it that we find asexual organisms. On the other hand, we also see an interesting increase in genera richness with with altitude, and of course also with precipitation scenarios. Okay, so we wanted to understand more about the ecology of these nematodes and find out how and if they can disperse at all, because this, you have to imagine, you see some rocks and some bushes and cacti, so there is a bit of life observable, but there is zero precipitation, and the only water availability comes from fog scenarios. Sometimes there's a bit of fog, and this is enough for these plants to survive. And obviously, we also find the nematodes, but the question is, how can they move? How far can they migrate? What are the dispersal patterns? And we did an analysis to investigate a plot, a migration experiment. Actually, we went along the taxonomic rank from the family down to the nucleotide level, and I just want to highlight some of these results. So, for example, investigating on the species level, what we found is there is some interspecific competition when we observe different habitat types, such as the cacti, the bushes, or the rocks. So when there's the one species, the other would not occur, and vice versa. So we see some interspecific competition. When we dive down to the haplotype level, in this case, this was 18S barcoding data that we applied first of all to get an idea on the species, and then also to understand the distribution of haplotypes in this plot. We also see this interspecific competition pattern, which is interesting because we see the same ecological pattern on different levels of biological organization. When we then wanted to go down to the nucleotide level, we had a bad time because we noticed, okay, the species that we need to sequence lacks a reference genome. This is the world of non-model organisms, obviously. So we had no reference where we could map our data to. And we were in lack of time because there was a master student who needed to finish. So there was not enough time to assemble one. And then we applied something, a novel strategy, analyzing so-called ultra-conserved element regions in genomes. So what is that? All the genomes, they actually contain highly conserved regions that are conserved across broad taxonomic range. So there is a set of ultra-conserved element regions, for example, defined for all the chordates, or actually for arthropods. And same is obviously also true for some groups in the phylum of nematodes. And we borrowed a core set of ultra-conserved elements from a distantly related nematode family. And what we found was, interestingly, more than 150 of these core elements we could also find in our species of choice here, Acrobeles. And of course, it doesn't make a lot of sense to investigate an ultra-conserved region for diversity patterns. What we applied is look at the flanking regions. And it was very interesting because we found, when we compared the diversity estimates from these flanking regions to diversity estimates from whole genome sequencing data of other nematode species, and also C. elegans or C. brenneri, so different Caenorhabditis model species, we found completely comparable thresholds and nucleotide diversity estimates. So obviously these UCE estimations produce a promising alternative to estimate genomic diversity in the situation where you do not have a reference genome at hand. And what we could study then is whether or not we find an evidence for isolation by distance, or maybe isolation by environment, because you remember there were the rocks or the cacti or the bushes. But what we found is this is not the case. So obviously there's ongoing gene flow between these individuals. And of course, the next step will be having the reference genome assembled and then diving into the estimation of migration rates as such. Okay, but in the beginning, I also mentioned this crazy life trait that these nematodes can enter this metabolic state of so-called cryptobiosis. This can be started by desiccation or by super high stress responses towards salinity, or by freezing, so cryobiosis. And we here investigate osmobiosis, so tolerance towards salinity, and anhydrobiosis, so the entire desiccation. And what we find is that anhydrobiosis and osmobiosis are variable traits. We tested different strains from the Atacama and found that most, if not all of them, can enter anhydrobiosis. Sometimes it does depend a bit on the environmental temperature that they are experiencing beforehand. And then most of them can also be rehydrated. And this is what you see down here. So basically, you see here these worms in an active state in panel B, and then they start desiccating, and finally they curl up, like in panel D, losing all of their water body, all of their body water, sorry. And of course, this makes only sense if they can be revived from this state. So rehydration is actually the trait that needs to be measured. And we found that it also depends on the environmental temperature, how successful this is. So this was very exciting and something we will dive into more detail. So what is actually the molecular trait of anhydrobiosis? it is expected to be highly polygenic. So we, in this case, decided to go not bottom up, but top down, and first start on a proteomics approach. So what we did is an experiment where we continuously dried up the worms, extracting them for proteomic sequencing. So then we passed the desiccation phase, rehydrated them, and got proteomics data from all these intermediate steps. And what we finally see is the protein composition does collapse, but can then be recovered more or less to the initial state that is resolved on this second axis of the PCA here. So you see the color coding of the time series, and through the desiccation process, you lose a lot of proteins, which then are recovered and reassembled upon rehydration. So this is very fascinating. And of course, it gives us a first indication on which proteins do contribute to the anhydrobiosis as a variable trait. And from here, we now have a starting point to dive into transcriptomics and, of course, trying to investigate the genomic underpinnings of this interesting trait. Yeah, I don't want to forget that these nematodes also vary in their reproduction modes, and we have been investigating also patterns or the impact of asexuality, parthenogenesis in the system. This was a study where we compared sexual as well as asexual strains from all over the world, and also estimated their mutation rates to see whether there's a difference between reproduction mode and the mutational evolution. And what we found is actually that one would expect that asexual strains that are cannot recombine and not deplete deleterious mutations, that they would not have an elevated mutation rate, but actually they range in the same magnitude as their related sexual strains. So the question is, how can they actually escape malus recet, so the accumulation of deleterious mutation here? And this is something that we will also look into more detail in the coming months. Last but not least, these nematodes, unfortunately, are in most cases, especially if they are asexual, they are polyploid. Here you see an example of a strain that comes from the Siberian permafrost, and it is triploid actually. So you see here in chart B, you see the 3 different genome copies, and it is very interesting because there is one genome copy that is very different to the others. So there has been obviously a diploid individual acquiring a 3rd genome copy via hybridization. And this resulted into the onset of parthenogenesis in this strain. So basically the asexuality is an acquired trait by incomplete hybridization and leading to polyploidy. So the question is, how can we understand the demographies from these genomes? So actually what we need to do is we need to look at these 3 different genocopies separately, reading out 3 different demographies and trying to understand the combination of different demographic histories that are integrated into one organism. Because for now, whatever we tried with existing approaches, we failed to reconstruct the genome-wide population history. Nevertheless, we are currently diving more into this to fine-tune the process of Markovian coalescence on the genome-wide scale, applying this to individual genome copies to resolve the demographic history here. Okay, so what can we then finally, hopefully at one point, when all the data is there, learn from genomes from this highly stable but extreme environment? Yeah, what we want to find is which genes are underpinning cryptobiosis and how does this trait actually evolve? How can genomic integrity be maintained if an organism is desiccated to 100%? So basically all these proteins, including also the genome as such, is falling apart in pieces. How can this be recovered, and what does it mean for the mutation load? And how would finally these periods that they sit into anhydrobiosis affect the signals for demographic inference? Because we have to make sure that we account for these periods in dormancy when we do population history inference. Do they pause evolution, or is the mutational process ongoing? These are all unresolved questions. And finally, what is very interesting also to learn how mutation rates can be controlled under asexuality. So basically, we face a super stable environment, so the mutation scenario should be constant, and we can here study sexual and asexual strains in combination. Okay, diving into the river part of my talk, a super dynamic system, the River Rhine here next door, back to the continent of Europe, back to Cologne. The River Rhine and its floodplain is the most dynamic stream habitat that I can imagine. There's constant flooding, frequent flooding events, frequent drought events. So we have everything that we can imagine in terms of habitat change in a year. So again, back to the question, how can we genomically assess eco-evolutionary interaction, especially in such a dynamic system? In this case, what we want to do is tracking genomic interaction on the level of biotic interaction in a food web. So obviously, the interaction of organisms in a food web should directly explain the mutual selection regimes that they exert on each other. One organism predating the other, obviously having an impact on the population size of the prey population. So you can imagine that there should be fluctuating signals on the genomic level. For this reason, obviously, we need to design a study that allows us to track genomic interaction on the level of food webs, what means we have to acquire a lot of data. And we do this in a long-term ecological research study that I have been initiating 2 years ago for the group here at the River Rhine, where we, on the one hand, do a lot of ecological monitoring. This focuses on the abiotic spectrum. And of course, we need to know which species are there and what is their abundance. And for that, we do apply a system that allows us to identify species and estimate their abundance using artificial intelligence and image recognition. And on the other hand, of course, we need to do whole genome analyses of different individuals, sometimes populations, sometimes even communities using meta approaches or relying on eDNA, depending on the different category in the food web. It is an enormous effort to acquire this data set, and we work across 5 different size classes. So ecology defines size classes to basically separate the universe of biotic interaction in an habitat into different size classes. And we have 6 different sites that we regularly sample every second month. We do accordingly 6 samplings a year and apply all these 5 different size classes, easily summing up to many, many samples in the time that we now passed over the course of 3 entire years. And of course, then we also need to identify the species, catalog them and sequence them, producing reference genome. And depending on the different size class, we have already achieved quite a reasonable status for the project. Long-term ecological research is nothing that we have invented, but it is an established network that exists on the international scale and accordingly also on the European level. And in Germany, we call this network LTRD, so the German Network of Long-Term Research. And there are currently 49 research sites established in Germany. And we are nowadays with our project RES, one of these sites, contributing to the understanding of freshwater systems. Our research site, as I just said, spans 6 different sampling sites, including the River Rhine as the mainstream, but also 2 oxbows and 2 gravel pit lakes that are associated to the floodplain. And it is a collaborative framework where many colleagues do contribute. We first had to assess the biodiversity in its status quo to learn who is actually there and how many. And so it was the first assessment across the different size classes and across the different sampling sites. A lot of morphological identification was involved, and then we applied some meta barcoding to get a first idea on the taxonomic profile. And what we could see is that actually the different habitat types, so stream, oxbow, and lakes, would be very different in their biodiversity. And what we also learned is that there's quite something that you can get out of meta-barcoding results that is hidden when we only observe morphotypes by morphological identification. To stress again that there's this need to implement molecular techniques into such assessments, because there's still many people are busy with morphological identification for biodiversity assessment nowadays. This is something I think we have to overcome. And here I only show you that we have, with only 1 marker gene, quite a good resolution across the different size classes, getting an idea on the biodiversity in our system. Nevertheless, we also have to see that in many cases, we cannot dive down, not even approaching the species level. So we actually don't get a very good idea on the biodiversity. And we have to say, barcoding cannot reveal the true level of biodiversity. Taxonomy in these groups that we are targeting in many cases is insufficient, even though we do research at our doorsteps right here in the middle of Germany, and our databases are super incomplete. So we have to face the scenario that we are a bit left alone in terms of genomic resources for our study system. What brought me to the decision that I really need to start something, a genomic initiative, bringing in efforts to standardize the generation of genomic resources for biodiversity here in our region. And this is what we founded, the Biodiversity Genomics Center Cologne. In short, this is BioC2. So what we have is some seed funding to generate high quality reference genomes on the level of an entire ecosystem. This is, of course, a collaboration with many partners here from the Sequencing Center, but also from Max Planck and our regional computing center. And Yeah, so what we hope is to bring in many reference genomes as quickly as possible to allow us to analyze the data that we are generating, because we all learned that genomics can be a powerful tool, not only for us being interested in particular basic research, but also in terms of biodiversity conservation. So what can we finally read from genomes that we get out of a super dynamic system, and trying to understand the ecology in these systems? So what I hope is that we can actually capture biotic interaction on the genomic level. Just imagine that one would not have to go out and assess a million of ecological factors in future. We would not have to do all this taxonomic profiling, but we would simply have to go out, applying some genome sequencing and getting an idea on what is going wrong or right in an ecosystem. But this, of course, also depends that we understand how strong this eco-evolutionary feedback loop between molecular evolution, the demographic impact in a biotic interaction, and the environmental conditions really is. So this is a question I have no idea. I'm super curious to learn from data that could, for example, look like this, that we have a time series of many interacting species across many different seasons, and then we, for example, track interacting allele frequency patterns. But as you all most likely know better than I do, this is an ideal world scenario. And as soon as we open the box of Pandora from genomes, we will learn that it is not that clear cut. Nevertheless, it's worth a try, and we will continue to collect a lot of data. And with this, I already close. And thank you for the attention. Thank you for ideas that you are very welcome to pitch, and yeah, for questions.